Hi, everybody. Thanks so much for joining us. We have a special guest today, Dr. Kevin Otto. We're also doing our webinar at a special time today. Our normal webinar schedule is every Thursday at 10 a.m. Eastern Standard Time and also at 2 p.m. And next week, we're going to be doing Beyond Probes, Engineering Your Experiments with our engineer, Peter Garreau. He's going to be talking about how engineering can make your experiment more robust, easier to perform, cleaner signals, and some of the things he's going to be talking about are custom chambers or custom drives. He'll give you some examples of things he's done in the past. And so look for the invitation on Monday. We'll send that out. And you can sign up for that. And with that, I'm going to introduce our speaker. Dr. Kevin Otto received his BS degree in chemical engineering from Colorado State University in 1997, his master's degree in bioengineering in 2002, and his PhD degree in bioengineering in 2003 from Arizona State University, Tempe. From 97 to 2003, he was a research assistant in the bioengineering department at Arizona State University, where his work in the areas of neural engineering and sensory neuroprosthetics. From 2003 to 2004, he was a research fellow in the Department of Biomedical Engineering at University of Michigan, Ann Arbor, where his work focused on brain-machine interface systems and implantable devices. From 2004 to 2006, he was a postdoctoral fellow in the Central University of Michigan, Ann Arbor, where his work focused on cochlear implants. Sorry, I just skipped a line. So 2004, 2006, he was a postdoctoral fellow in the Central Systems Laboratory in the CRIS Hearing Research Institute in the Department of Orthonology at the University of Michigan, Ann Arbor, where his work focused on cochlear implants. He is currently a professor in J. Creighton Prout Family Department of Biomedical Medical Engineering at the University of Florida. His research interests include neuroengineering, device tissue interfaces, and neurostimulation. And with that, I'm going to hand it over to Kevin.
Thank you very much, Matt. And thank you to NeuroNexus for the opportunity to speak in the webinar. I really appreciate the fact that you're doing this during this time. It's been a great opportunity to learn more about NeuroNexus, as well as a good opportunity to be able to speak with colleagues and talk more about science. So today I'll talk about our work in my laboratory, detailing a lot of implantable devices, primarily in the brain. And a lot of these devices have been NeuroNexus devices, or the Michigan probe before they were NeuroNexus devices. So let me jump into it. What the rationale for understanding these and the motivation really stems from some of the exciting opportunities that have come out of work using implantable devices, primarily in cortex of human patients. In this case, this individual was implanted with a Utah electrode array, officially from BlackRock Microtechnologies. She was implanted in her primary motor cortex. Prior to that, she had suffered a brainstem stroke, and she was completely quadriplegic and unable to even get a drink of water, or in this case, coffee. She really enjoys coffee. So what she's thinking about doing is thinking about moving her arm, and the arm movement part of the brain, part of cortex, is active. It's working. It doesn't explicitly know that the communication is not getting through to the muscles. as they record that activity, it's decoded by computer algorithms and turned into the articulation of the robot. And that enables her to successfully reach and grasp and drink from this carefully product placed mug that she's drinking her coffee out of. And the exciting part about this is that it's voluntary. That's the first time that she has been able to, on her own, take a drink of coffee since her cerebrovascular accident. And I think you'll see here in just a second, she gets a lot of enjoyment from that aspect. It increases her quality of life quite a bit, just accomplishing a simple task that many of us take for granted. Some of the disadvantages of this approach, one, you can tell it's pretty slow. The overall information rate that they're pulling out of the brain is nowhere near what you have with natural movements. Two, there's some noise in the decoding and the achievement of the movement. It's not a smooth, continuous movement. And three, she had to focus very intently on the movement of that robot. And most of the patients remark after sessions like this that they're exhausted. They are cognitively exhausted from working so hard. And we believe that part of that exercise that they're performing, part of that mental drain has to do with the mismatch of sensory information coming in. The only way that she knows where the robot is from looking at the robot. And that's not how typical movement is achieved. Most individuals can reach and grasp without looking at their arm. And they're able to do that through natural sensation that comes in through proprioceptive and touch sensors. So in this next video, this individual,
I don't know what we got in the point where we're going to do this one. Let's see what he got. Boom. Just blow it up. You want to blow it up or? That is unbelievable.
Robot. And so that's a significant step forward in terms of not only recording data from the brain, but also writing data into the brain. So what's limiting this? Going to the clinic and being implanted into spinal cord indentation and brainstem stroke patients and ALS patients. The simple reality is that it's not the robots. The robots are pretty phenomenal. Example from Applied Physics Laboratory at Johns Hopkins University. And you'll see that this robot can do just about everything that a human arm can do. And in some cases, it has clearly more flexibility than I need. And between the mechanisms, calculation sensors, it's vast. So the robots are not the problem. What has been the limiting step so far is the impedance. What you can see though is the robots are phenomenal. The robots are worth going forward with. What the challenge is that the individuals are not able to use the implants for a long term. That you put the implants in and they work really well in the beginning. And typically over the course of days to weeks to months, they gradually tend to lose a little bit of performance. And this is not necessarily novel to brain implants. This is something that shows up with implanted sensors and electrical actuators in the body all the time. I'm type 1 diabetic and use both implantable glucose sensors as well as implantable pump conduits, and in both cases I have to change those out every 3 to 10 days because of the same limited lifetime. And that's a major challenge for brain implants. Changing out your brain implant every 3 days is not feasible at this point. So what we need to do is figure out how to improve the overall lifetime of these devices so that we can actually get them to be used in the clinic long term. And I am actually going to jump ahead since we're a little bit behind. Today, I'm going to talk about our lab's work on brain interfaces specifically, trying to evaluate them, assess them, as well as our efforts to improve them in some ways. In my lab, we also do quite a bit of work with peripheral nerves. And in both cases, we look at autonomic neural engineering and how that can be applied to the blossoming field of bioelectronic medicine. But that's not going to be the focus of today's talk. We're simply going to talk about brain interfaces today. So I thought I'd give a brief introduction. Most of the people on this call probably don't need an introduction to this device. These are all different versions of and different zoom, optical zooms of a neural nexus device. In the top, you can see an acute device that is not mounted to the connecting board, not mounted to the printed circuit board. This is the same device mounted to that printed circuit board, ready for acute implantation and experimentation. Here's a version of a chronic device where this can be implanted and sealed up so that you can connect and disconnect from your subjects when they're either home sleeping or away from the laboratory. And if we zoom in, you can see here's an example of little tiny electrode sites that are spaced on very even intervals going down the surface of one of these devices. So this is a very common approach that we would use in our laboratory. Periodically spaced linear sites. We oftentimes use 100 micron spacing, and the site size will change depending upon our application, but I'll try to mention that as we go forward. We have done quite a bit of work on looking at the electrochemistry and the actual electrolyte interface, especially with the application space of stimulation. We're trying to apply electrical currents to the devices and excite local activity from the cells that are situated near the electrode. One of the challenges there is to control the electrical energy so that you don't result in either electrode degradation or tissue damage local to the electrode. And in order to do that, one aspect that matters in that whole equation is what the chemistry is of the electrode-electrolyte interface. Changing the chemistries from blank metal devices like iridium devices or gold and oxidizing those surfaces can help to create surface area. It can also create a substrate for Faradaic reactions that can enable charge transfer. So in this case, published in 2009, we took iridium devices and we oxidized the surface of the devices to increase the charge carrying capacity. We also put down a conductive polymer, polyethylene dioxythiophene, which tends to form this little mushroom cloud on the electrode devices. And you'll see throughout, I'll talk a little bit about the surface chemistry, though we won't dwell on it too much. But in that paper, if you're interested, we detailed how these different materials enable different stimulation currents and the subsequent voltages needed to push the currents that we were interested in. Once the surface chemistries are identified and characterized, we implant the devices. We oftentimes do a permanent implantation for chronic longitudinal data collection. So in this case, this is a device that's been implanted. This is in the primary auditory cortex of a rat, and this is ready for cement that's going to cement everything into place. And here's a final product that you might see where we have an animal who's been implanted. He is happy and healthy, and you can see the connector there on top of his head, so we can plug in when we are ready to either record or stimulate. And in fact, this animal is at his water spout drinking water, which I'll come back to in a while. We do a lot of behavioral experiments, asking the animals questions, and one of their response metrics is their behavior of drinking water. So one of the downsides of putting the electrodes into the body is that the body doesn't like to have outside activity into its carefully protected spaces. And of all the carefully protected spaces, the brain is probably one of the most protected. There's a lot of mechanical protection with the skin, bone, and dura, but also there's very tight chemical and immunological protection spaces within that area. So as you implant, you do mechanical damage on the insertion. You also have materials that are foreign, that are identified as foreign, and both a resident immune response as well as an activated immune response that tend to encapsulate the device, and ultimately they try to eat it, they try to digest it. These cells, when they realize that they can't actually digest silicon, for example, they go through what's known as frustrated phagocytosis, and they start to emit a lot of signals, cytokines, etc., that can result in a neuronal cell loss area. And it's not completely clear if it's actually neuronal cell death or if they're being pushed away by the glial cells that are coming locally. But oftentimes when we look at chronically implanted devices, you will see this area where the neuronal population is lower than it is at distances some away from the electrode. We also, if we look at that electrically, we see that as an impedance increase. What that means is if we measure the complex resistance and reactance between this electrode site and a distant ground, these cells create an ionic barrier to current so that the electrode is no longer able to communicate across that wall as well as it could in just basic saline or otherwise. And we think that overall, all of this put together leads to an efficacy decrease. And we see that both in recording and stimulation approaches. So when we actually try to characterize this, one of the things that we'll look at is implanting devices of various aspects, putting them in and recording or stimulating, measuring behavior in the stimulation case, quantifying the electrophysiology recordings with their overall performance, and then doing a post-mortem evaluation, histologically looking at the device and the tissue, and hopefully trying to look at those in concert with each other to see how we can close the feedback loop of designing the devices, implanting the devices, sterilizing devices, and using them, programming them effectively. So what's the use case? What's the data that the devices do not perform as well as we'd like? This is a publication by Neuronexus's own Rio Vetter from 2004, looking at 9 different rats that were implanted over the course of 18 weeks. Some of the animals were terminated early, usually due to head cap failure or other aspects of failure of the implants. And overall, what you can see is that the signal to noise ratio of the overall performance of these devices is going down over the course of 18 weeks post-implant. 18 weeks is a long time for some science experiments. If it was your brain that was being implanted for spinal cord injury enabling movement, 18 weeks is a little more periodic than most people want to undergo brain surgery. So this is an admirable performance and was a great paper detailing that performance, but we do need to improve the slope of that line. And just to reiterate that it's not Neuronexus or Rio or anything in particular to do with that paper. This is an example of a different animal model. These are guinea pigs. This is a different electrode model. These are tungsten microwires. And in this case, you can actually see 2 different versions of the same line. Some animals lost their devices almost completely in just 15 weeks, and some did much better, but you can see that the performance is still decreasing. If we look at it in our data, this is data from my laboratory that's just emerging, and Morgan Urdaneta has collected a very comprehensive data set. And one of the things that Morgan has really tried to focus on in these experiments is how does this matter with cortical depth? We have access to 16 different depths with a typical linear 16-channel electrode array. And if the electrodes are designed to span perfectly across all the layers of cortex, spanning from layer 1 all the way down to layer 6, we have the ability to look at performance of each layer as a function of time post-implant. So here you can see raw electrode signals versus filtered electrode signals across the depth of the electrode array. The colors match up to the layers, as you see here. Morgan has gone through and quantified, and you can see here the mean spike amplitude typically is greatest around layers 4, 5, 6, typically centered close to 5. And in fact, the box and whisker plot, you can see that that peaks at layer 5 as well. You'll see sort of repeatedly, that's a theme throughout this data set, is layer 5 is sort of a very good target for optimal performance. If we look at this with time post-implant, this is spike waveform amplitude across the different layers, and you can start to see differential effects, especially changing. Morgan has decided that at 8 weeks post-implant, there's a shift in variance and overall performance. And so trying to average the first 8 weeks versus the second 8 weeks, one of the things you can see is that the amplitude effects start to differ a little bit as you can change this, but the overall profile is still there. Layer 5 is still the highest amplitude in the first 8 and maintains its best performance across the layers as the weeks go out. If we look at a different analysis, which is the signal to noise ratio of these, here you can see our sort of characteristic line decreasing over the time of 16 weeks post-implant. But if we look at the weeks or the layers individually, the one long-term that's still performing the best is still layer 5. So that still tends to be the layer that should be targeted probably long-term for, in this case, a recording application. They also remain the most active over time. Over the course of time post-implant, right in that layers 4, 5 sweet spot, you can see those are the channels that are active the longest through this analysis. So if we switch from a recording paradigm to a stimulation paradigm, we can ask the same questions. Is the performance affected by time post-implant? How does the performance differ across layers? Are there other aspects that we can achieve to quantify the performance of stimulation? And so one of the ways that we evaluate stimulation efficacy is through this behavioral metric called conditioned avoidance behavior. The animals in this case are it's very much like an electric dog fence. We have one at home, so I'm comfortable training my animal. We have a dog, we take him out there in the beginning. He doesn't have any clue about the fence. He has a collar on that emits a sound and warns him he's getting close to the fence. When he actually gets too close, he gets a little shock, and very quickly, we have a golden retriever, so he learned it the first time that shock or sound means shock and don't go there. So the animals are doing the same thing. They come up to the spout. It looks just like their home spout at home, and they start drinking water. We start pumping water to them. We're measuring their licks. As you can see on the back, they're initially trained with an acoustic sound that's emitted from that speaker. And again, that sound means their shock is coming. If they're still touching the spout, we give them a small electrocutaneous shock on their tongue. I personally haven't done it, but I've had a graduate student who did it, and he said it feels like a 9-volt battery on your tongue. It is not pleasing, but it's not damaging. It doesn't hurt, and it doesn't dissuade any future drinking at all. But they learn pretty quickly to avoid it if they can. And so if they hear the sound, they will stop licking, and you'll see actually the animals are incredibly good at this task as we watch this movie. And then what we do is we gradually turn the volume of the sound down to try to find the threshold. Once the animals are good at the acoustic task, we implant a device and into their sensory cortices. We initially did all this work in auditory cortex, and we've since learned that it works in somatosensory cortex. It also works in visual cortex. The animals can make the association across sensory cortices, no problem. And as soon as the animal feels this acoustic or auditory cortex stimulation or somatosensory cortex stimulation, they stop licking. So in this movie, what you'll see is that the blue represents the lick patterns and the licking. The animal would begin licking, and then the yellow will depict the stimulation. And so your job is to either watch the data set to see when stimulation is delivered, if the lick pattern changes, or you can watch the corresponding video to see if the animal's behavior changes. So the lick patterns are recorded. Here, we slow it down so that you can watch the behavior closely. We're going to deliver the stimulation. It's on right now. And if you were watching closely, you notice that the animal pulled back and stopped licking. You can also see that in the data shown here. And so with this, the animal successfully told us that he felt that stimulation. And since he felt it, the next time we're going to deliver a little less current. And so we turned down the amplitude a little bit. And you'll see he actually got that one correct as well. He detected that. And so the next time, we'll actually turn it down and turn it down. And we'll keep doing that until we find threshold. One of the advantages of that task is that it's very fast to determine a threshold on an individual channel. It's also very repeatable. If we do it on the same channel multiple times in a row, we get a nearly the same answer across multiple trials. So it's an optimal task for this kind of behavior, where we want to come in and we want to assess 16 different stimulation thresholds, perhaps multiple times on a given day, and then we want to send the animal home and bring them back tomorrow and ask the same question. If we look at the performance over time, we're going to quantify that by the threshold amplitude. So over time, if the animal is getting less and less sensitive to that stimulation, then we would expect the thresholds to go up. Because we have access to the different levels of cortex, we're going to analyze the most shallow, the intermediate zones, and the most deep zones. So we have the axes of depth in cortex and time post-implant. And versus those two axes, we plot the threshold, and you end up with this graph. Here is the depth. Here is time increasing as you get lighter and lighter. And so two take-home messages here. First of all, at any time, deeper layers typically have lower thresholds. At any depth, threshold typically increases over time. And those two axes are heavily dependent on each other. Those lines are parallel, are not parallel. So if we actually lump the data into these four different groups that you see down here, you can see that the more shallower sites are increasing threshold faster. The deeper sites seem to be much better, and the overall average is somewhere in the middle of getting worse, but not at such an increasing rate. We have since, Morgan Ordinetta has gone back and really built on this data set here at the University of Florida. publishing this again a little more robustly with the layer discrimination. And also, one of the remarkable things about this data set is the time that he's been actually collecting. So this goes out to 40 weeks, which significantly extends our previous experiments that we published. So it's the same takeaway message, actually, that layer 5, first of all, is the most stable. And secondly, is the most reliable. It actually gives you the most active channels that we didn't lose over time. You do see this remarkable aspect. If you overall average all of the data, you start to see that same difference between the first 8 weeks versus everything after the 8 weeks. And in the first 8 weeks, the stimulation, actually the thresholds go down overall. And then post 8 weeks, they go into this chronic phase where they go up over the long term. So this was shown at SFN last year, and is also, Morgan is rapidly working on putting these publications together. So we'll shift gears and talk a little bit about the histology here for 10 minutes or so. One of the things that we published in 2011 is actually the figure that I use as my background, and that is this device captured histology approach to trying to understand what is it about that interface that could be changing, that could be resulting in these effects that we're seeing. In this case, this is a NeuroNexus device that has been implanted into the rat's brain. I don't know why the investigator chose to put it in at such an interesting angle, but it does sort of set up for a nice picture where the device is contrasted against the tissue. For reference, here's a blood vessel on the right-hand side, so you can see the scale bar there at 100 microns. The overall electrode is about 100 microns across. These are rectangular electrodes, so it's very skinny in the Z dimension. And consequently, the investigator, Andrew Woolley, was able to slice the tissue parallel to the device. In order to do this, he had to take very thick slices. This is a 400 micron thick slice, but that captured the entire device in place. What he's then able to do is, using very careful and iterative mechanisms to stain the tissue and then image the tissue, he's able to look at multiple different stains inside of the same sample to try to investigate what cells are participating in these kinds of responses. So here in this case, he stained for microglia. If you hadn't seen on the last, before the animation, this device was only implanted for a week. So we start to see some microglial activity near the surface of the device. It's definitely more towards the surface of cortex. But overall, this isn't a really large response at 7 days post-implant. He was also able to look at astrocytes. You can see astrocytes doing their normal function of helping with the blood-brain barrier, both at the surface of cortex as well as around this blood vessel. They're also distributed throughout the brain, helping to take nutrients out of the blood vessels and deliver them to the neurons, as well as do some synaptic modulation and all the other aspects that the astrocytes do. And ultimately, they are starting to change morphology near the electrode. If we were to look at this much later, you'd start to see this kind of layer being built up around the electrode of limiting the building up that actual blood-brain barrier, or device-brain barrier in this case. Then finally, Andrew was able to look at oligodendrocytes in this case. And so oligodendrocytes forming the myelin around the axons. You can see a lot of normal cellular architecture, cortical connections here, columnar connections here, different cortical columns you can almost envision in these different spaces. And interestingly, you can see, first of all, a lot of disorganization surrounding the device, but also an overall lower amount of myelination, a lower amount of signal there. potentially indicating a demyelination event or progression with the device being in place. So we've gone on to develop other techniques to enable this as well. And one of the challenges of doing the device capture histology is, first of all, it sometimes it's a bit risky because the devices fall out oftentimes during the process. It takes a lot of skill and patience and luck to get that kind of picture. So an alternative approach that Morgan Urdaneta has been approaching is building this pipeline where he can do an analysis of the histology with depth of the device. And he's brought in different machine learning techniques to enable some of the analysis. In this case, the device is explanted, and then careful record of the depths post-implant are kept with along different slides. This is again being prepared for preparation or for publication. But ultimately what this leads to is an enabling of quantification. In this case, he's going to look at neurons and cell loss using these machine learning algorithms to identify the volumes of cell loss in each of these individual slices. And one of the interesting things that he's found with his data set is that the most cell loss corresponds to layers 2, 3, and even layer 1. And the layers in 5 and 6 have not only lower loss, but layer 5 again is kind of the winner. So not only does it win in the histology battle, it wins with the recordings and the stimulation. Again, giving us a lot of excitement that we're starting to hone in on exactly the right area. If you quantify that with these violin plots, you can see this is the same data set sort of turned on its edge and analyzed, shown comprehensively with these violin plots, showing the layer 2, 3, with the higher loss areas and layer 5 being the lowest overall. As an additional alternative, we've sort of extended the previous device capture histology, and we're trying to do this not only in 3 dimensions, but also with device clearing and light sheet microscopy. So in this case, this device was left in place in this 3-dimensional section, and you can see the device residing in that tissue, in tissue that's been cleared so that you can see through it. And what this enables is with we use clarity as our clearing mechanism, and in this case, we labeled the device or the tissue with CNPase, which is a marker for myelin. And you can, we're still continuing to work on this and developing the protocols, the antibody labeling, as well as the imaging and ultimately the analysis protocols. But these are the kinds of approaches that are being enabled with this volumetric 3D, and Morgan likes to say 4D because we're including the labeling of the individual cells as well. So we're adding dimensions in many ways to the data set. One more additional histological approach is to actually look at all of this in vivo. And that enables the longitudinal aspect of approach. So if we're able to label the cells adequately and accurately and implant the devices so that they can be imaged, we could try to observe the response of the brain and the effects on the electrode as a function of time post-implant. And so in order to do this, we have adopted the window technique of replacing the skull with a window, a glass window or a constructed window that's clear that you can see the device implanted. In this case, because of the imaging limitations, we implant the device at a somewhat oblique angle, a shallow angle. So we're only looking at layers 1, 2, maybe 3. And this is what it looks like under the microscope. This is just a light microscope. You can see the device actually going underneath these blood vessels here. One of the interesting things is that most of the antibody approaches that you use in immunohistochemistry are all toxic in vivo, so you can't use them for actual alive animals. So in order to label the cells, we've adopted genetic techniques of using green fluorescent protein. And the first case that we did was we labeled a mouse with EGFP and asked the microglia to express that protein. So in this case, looking underneath a fluorescent microscope, you can see that this brain is glowing green as all these little microglia are fluorescing with their EGFP. If we look at that under multiphoton, you can start to approach single cell resolution. You can see the individual microglia hanging out around this electrode device. And so if we add the one more dimension of time, this is a video that is going to start to scan down the device. And this is an implant at 2 days post-implant. So the device hasn't been in there that long. This is about the point where the device enters the brain, and then it gradually slopes away from you, going down this way. So we're basically turning down and going deeper into the brain with the depth of focus. You can see the outline of the device tissue interface right there, some shadows of blood vessels throughout, some weird cerebrovascular accident up here that we didn't take any credit for. But overall, at 2 days post-implant, this doesn't look too bad. How about 4 days later? 4 days later, again, you can very clearly see where the device enters the brain now. And it's pretty easy to see the outline of the device as all of these microglia who have either proliferated or migrated or probably both, and they're activated, they're aggressively attacking the device at only 6 days post-implant. So in order to extend this, we really need to look at not just GFP and microglia, but the other cells of interest as well. And so in this case, Janak Iyer 2 years ago published a paper where we constructed a mouse that has 4 different fluorophores that identify microglia, oligodendrocytes, neurons, and astrocytes. If you're interested, there's many, many cool images in this paper that Janak was able to put together. Here's examples from cortex and cerebellum. He also in that paper detailed the device tissue interface for implanted animals with like this, but I'm not going to talk about it here. In brief, we've used this overall data to start to design approaches that we can either change the device design or add in things like coatings or drug release or even electrical aspects. And so in brief, we've actually attempted many different aspects of trying this and are in process of developing several different strategies to try to address the curve, try to get that curve back up to flat performance. And a couple of ones that we've published a little recently that I thought are worth highlighting here. Caitlin Olzak in my laboratory has published a paper in ACS Applied Biomaterials where she used layer by layer coatings that were applied to the electrode. And in this case, she looked at releasing minocycline from these devices. And she also characterized the electrical activity of these devices to show that the devices weren't negatively affected by the layer by layer coatings. We've also done quite a bit of work looking at electrically getting rid of the glial encapsulation. And so work that I did as a postdoc in collaboration with Matt Johnson while we were at the University of Michigan was to ask if we could use electricity delivered via the microelectrode to get rid of the glial encapsulation. And I won't belabor this too much, but one of the things that we found is that we were able to recover electrophysiology performance on selected sites. So we would apply these electrical potentials to the sites and rejuvenate them, recover some of the performance. It didn't work for all electrodes all of the time, but in a subsequent study that I did as a postdoc in John Middlebrooks's lab, we collected longitudinal data from guinea pigs where we were measuring the impedance of the device and measuring the electrophysiology performance of the device, and then rejuvenating the electrode sites, subsequently performing the impedance and the recording again. And so in this case, what you're looking at with these data are the longitudinal effects of the impedance magnitude before and after that electrical rejuvenation. And here's the corresponding signal to noise ratio, where here's the before performance going down and not really that good. A SNR of 3 and lower is pretty miserable. But in all cases, as we rejuvenated the electrode device, the impedance went down and the signal to noise ratio went up. And on these same devices, when we brought them in day after day, you can see that overall, every day we had an improvement. It is important to note that this overall curve is still decreasing, and it was no silver bullet that fixed everything, but it did significantly improve performance. One last thing that can improve performance, we've published this in 2017, is you move the sites to the edges of the silicon body. So in this case, we compared sites that were in the middle of the electrode, the tip, and at the edge. And we did that on 2 different body width electrodes. Based on the model shown here that many others have actually developed, including Patrick Tresco, we predicted that the electrodes on the sides should perform overall much better than these electrodes in the middle. And in fact, that is what we saw, is that the electrodes that were in the center were overall had a lower number of active electrodes, as well as, and that worked regardless of wide or narrow electrodes. And that was true both in the shorter week, the 7 days post-implant, as well as out to 7 weeks post-implant. One of the other sort of interesting aspects that we looked at, and there's some caveats to this study, but we tried to look at the role of inflammation, thinking that if it is this inflammatory model, that maybe the inflammatory cytokines are contributing to this decay, maybe we can actually upregulate inflammatory cytokines to see if we could cause the devices to fail faster. And so what we did in this study is we used LPS, which is a pro-inflammatory molecule that aggravates the microglia specifically, and we know causes an increase in microglial activity. And one of the things that we saw, if we look at TNF alpha, is in the very beginning, we actually saw TNF alpha effects with the LPS, even if we were trying to quell that with dexamethasone. We saw effects of the TNF that went away, but overall that actually didn't affect the device performance in these metrics that we looked at. This is a performance of signal to noise ratio. We also looked at impedance measures and other measures of electrophysiology performance. So there are some caveats, as I mentioned. First of all, this was only done with systemic injections, and we don't know exactly how much made it across the blood-brain barrier and how much, though we did do histology, we don't know how much of that pro-inflammatory signaling was really local. So this could be repeated with local drug delivery to see if how spatially confined the inflammation needs to be. So in summary, I would like to highlight the members of my group who actually do all the work. I'm just lucky enough to go out and tell as many people as I can about it. In particular, Janak Gayer, who is the individual that published that last paper, and Morgan Urdaneta is here right behind him. He collected much of the stimulation recording work that you saw earlier. I also showed work by Kaylin Olzak here.
All right, that was fantastic. Thank you very much for your time, Dr. Otto, and thank you everyone for coming. Watch Monday your email boxes to be notified about our next presentation. Thanks so much.
Thank you, everyone. Appreciate it.